Hello everyone, welcome back to BioBlinkers. In today's video, I'll be discussing about the transcription in prokaryotes. So, transcription basically means copy. In most of the organism, DNA is the genetic material. When the information is copied from the DNA to RNA, this process is known as transcription. When the information is transferred from the RNA to the protein, this process is known as translation, transcription, translation, replication. All these three processes comes under the central dogma of biology. Transcriptional unit basically consists of three components: promoter, terminator, and the structural gene. Promoter is the site where the transcription starts, and terminator is the site where the transcription stops. You know that DNA is a double-stranded structure. It consists of two strands, coding strand and the template strand. During transcription, RNA is synthesized using the template strand. If you look at the sequence of the coding strand and the RNA, you will observe that the sequence of coding strand and the RNA is exactly similar. The only difference lies is in the presence of uracil instead of thymine. Otherwise, the entire sequence is same. That is why we use the polarity of the coding strand to define the position of the promoter and terminator in the transcriptional unit. The polarity of the coding strand is 5' to 3'. So, the position of the promoter is 5' upstream of the structural gene and position of terminator is 3' downstream of the structural gene. Upstream means ahead of the structural gene and downstream means behind the structural gene. In prokaryotes, the two processes, transcription and translation, both occurs in a single compartment, that is cytoplasm. In prokaryotes, the transcriptional unit is polycistronic. Poly means many and cistronic are the alternative name of structural gene. Here you can see a single promoter is transcribing all the three structural genes A, B and C. That means in prokaryotes, the transcriptional unit carries information for more than one structural gene. The best example of polycistronic transcriptional unit is lac operon. All the three RNA, mRNA, rRNA, tRNA all are synthesized by a single RNA polymerase in prokaryotes. The RNA polymerase is basically consists of 5 subunits: alpha, beta, beta prime, omega and sigma factor. Alpha, beta, beta prime and omega known as core enzyme. Core enzyme and sigma factor together known as the holoenzyme. These subunits perform different functions. Alpha is involved in assembly of core enzyme. Beta and beta prime is responsible for the elongation of RNA strand. Omega is responsible for assembly of RNA polymerase and sigma factor is known for promoter recognition. Now let's discuss the process of transcription. Promoter is a segment of the DNA where the RNA polymerase binds and the transcription is initiated. Promoter consists of the conserved sequences. Sigma factor of RNA polymerase is the one which recognizes these conserved sequences and allow the RNA polymerase to bind at the specific site in the promoter. When the RNA polymerase binds with the promoter, transcription is start and this process is known as initiation. Now, a transcription bubble is formed in which the two strands of the DNA are separated and a small stretch of mRNA is synthesized using the template strand of the DNA. The transcription process is governed by the principle of complementarity. Now, when this mRNA chain reaches 8 and 9 base pair, the sigma factor of the RNA polymerase is released and the other subunit of the RNA polymerase, that is beta and beta prime, start adding the incoming nucleotide to the mRNA chain and this process is known as elongation. One important point that the synthesis of mRNA always takes place in the 5' to 3' direction. In DNA, some termination sequences are present When the RNA polymerase encounters these termination sequence, the rho factor starts stalling over the mRNA until it encounters the RUT site in the mRNA. Once it encounters the RUT site, the rho factor cleaves the RNA and DNA complex and mRNA is released. This process is known as termination. Now it's time for some facts. Did you know? DNA is composed of two components, exon and introns. Exon consists of the coding sequence and introns are non-coding sequence. Exon codes for a functional protein and introns does not code for any protein. Removal of introns is known as splicing. In prokaryotes, DNA lacks introns. That means splicing mechanism is completely absent in prokaryotes. If splicing is absent, that means the post-transcriptional modification will not be seen in prokaryotes. Now, one most important point, transcription and translation is coupled in prokaryotes because both process is taking place in cytoplasm. Thank you for watching this video. If you like this video, please subscribe to my channel and don't forget to like, share, comment and hit the bell icon so you don't miss any notification. Thank you and see you in the next video.